One important thing that cells communicate to each other is the need for making more cells, or cell reproduction. So cell division has two functions. There is a need to grow and make more cells and in the maintenance and repair of tissues. There is also a need for an organism to reproduce itself and therefore produce more cells that accomplish that task. In both of these cases, we will make use of the genome, which is where we find all the DNA found in the cells. So if we take a look at the different genomes that we find in prokaryotic versus eukaryotic cells, we'll start with prokaryotic, or the bacteria. So the genome for bacteria is the more simple of the two. It's a single double-stranded circular DNA molecule. And this resides not in a nucleus or a special container, but rather in a region of the bacteria called the nucleoid. So there's no membrane around this DNA. There may be additional smaller pieces of DNA that are called plasmids. These are also circular DNA. And they're not necessary for normal existence, but they may carry important information that can be exchanged between one bacteria and another. So they can swap these plasmids between each other. Now the eukaryotic genome, so remember the eukaryotes are the protists, which are mostly single-celled organisms, the fungi, plants, and the animals. They all have a much more complicated genome or set of DNA. This consists of several, depending on the species, double-stranded DNA molecules in the form of chromosomes. And we know that, for example, the human chromosome has a total of 46 chromosomes in the genome, 23 pairs. But this number will be different for each species. So as I mentioned, that when we look at eukaryotic chromosomes, they're present in the cell nucleus, they're sequestered in the nucleus, and it varies. The number varies depending on the species. Humans have, for example, 46 total chromosomes, 23 pairs of them. Here's the 22nd pair, here's the 23rd pair, which are the sex chromosomes, where we look and we see that in Drosophila or flies, there's only 8. And in dogs, there's 78. In potatoes, there's 48. So the number of chromosomes does not necessarily indicate intelligence. Within a species, though, that chromosome number is the same in most cells. So you notice that these pairs of chromosomes, pair 1 through 22 in the human, these are called autosomes. And the last pair are what we call the sex chromosomes. So in somatic cells, this is where we have typically 2 matched sets of our chromosomes. So we have 2 copies of every chromosome. That's why they're called diploid. Di means 2. And we represent that as a 2N. So in humans, we have 23 pairs of chromosomes for a total of 46. So for chromosome pair number 1, you have 2 copies of that. One you received from the maternal side, one you receive from the paternal side. Now, those somatic cells are the majority of the cells of the body, but in the gamete cells, these are the eggs and the sperm cells, there is only half the number of chromosomes, or 1 complete set of every chromosome. So haploid meaning half, So human egg cells or oocytes and sperm cells only have one copy of each chromosome for a total of 23. So I ask you, why do you think they only have one copy each? Now the chromosomes can be arranged by size, and this produces a picture, which is called a karyotype, a picture of the chromosomes. Chromosomes or homologs that pair in the reproduction of diploid cells are called homologous chromosomes. So here, for example, chromosome 1, one copy of it, chromosome 2, here's another, the second copy of it. When those two pair up, they are called homologous. So one came from mom and one came from dad, and they're present in every cell of your body. Some genomes have pairs that don't match. For example, the X and the Y chromosome in humans. So, and this only occurs in males. So for, say, chromosome 1, this chromosome matches this chromosome for the meaning that at every location there are alleles that code for certain traits, and you'll have the same allele in this spot as you do in this spot, all the way down the chromosome. But when you get to the sex chromosomes, you notice there's an X chromosome and a Y chromosome, and these do not have the same genes that match up. So those are called heterologous pairs, where they're different, hetero meaning different. The eukaryotic DNA is very large. It's quite a large molecule, and so we have to be able to compact it in order for it to fit into the nucleus. So here we're looking at just a tiny little piece of the DNA. Here's the DNA double helix, so we're looking at what it looks like, but this is only a tiny bit of it. It's quite a large molecule, and it gets wrapped around something that's called a histone, or a series of proteins, and then you have multiple ones of these. So this is one DNA wrapped histone, and look, each one of these is a histone. So we have to take these histones and in their string and coil those into nucleosomes in order to get them to fit in. Then there's further condensation once we get into the duplicated chromosome. So here's just one section of a duplicated chromosome. So these really, really need to be compacted in order to fit into the nucleus. So short stretches of DNA wrap around a core of 8 histone proteins, like this, like a string of beads. Then the histone DNA complex is called a nucleosome, and the connecting DNA is the string. So this is a nucleosome, and here's the connecting piece. That would be that string is called the linker DNA. And this structure coils to form the chromatin fiber. And then fibrous proteins further pack in order to condense the material even more. So here is a short YouTube video which will go into further detailing of chromosome packing. All right, now that we have a better understanding of the genome, or the DNA, we're going to talk about the life cycle of cells and how the DNA is going to play an important role in that. So when we look at the life cycle of a cell, we call it the cell cycle, and there's a specific order to the progression through this cycle. So we see that we are going to start the cycle primarily here in what we call G1. We're going to progress from G1 to S to G2. And all of this purple band here all together is what we call interphase. So the majority of the life of a cell is spent in interphase. At the end of interphase, the end of G2, we're going to enter into a division phase. So we have mitosis and then cytokinesis. So they make up the mitotic phase, these two steps together. Mitosis is when we divide up the chromosomes in the nucleus, and then cytokinesis is when we actually divide the cell into two. And then we now have two daughter cells which will start each a new G1 phase. So let's take a more close look at the interphase first. So we have two major phases. The interphase, which is shown here in in blue, excuse me. The first part is called the G1 phase or the gap phase. So interphase in blue is the normal growth and preparation for the cell. G1 is the first gap phase. There may be a little sideline called G0, which is a resting state, but after G1, the cell is going to progress into the S phase, and it's during S phase that the DNA is going to be copied. So synthesis is to make. We're going to make a copy of the DNA so that we'll have sufficient copies of the chromosomes to be able to divide up here. At the end of S phase, we then enter into another growth phase called G2 or gap 2, and then we can progress into the mitotic phase, which is shown in red, in which that DNA that we copied way back here is going to be split up, and then eventually we're going to divide the cell. So interphase has three parts. The G phase, or the first gap phase, you don't see any real change, but the cell is biochemically active. It's doing the job that the cell was intended to do. It's doing the life work of the cell. The S phase is then the next phase that's entered into, and this is most important for making identical copies. This is like running the chromosomes through a copy machine, and we will hook together the like copies. So every time we take a chromosome, say the maternal chromosome of chromosome number 1, we're going to make a copy of it, but we're going to stick those two copies together, and we're going to call them sister chromatids. And we're going to join them together at something called a centromere. The centrosomes produce the mitotic spindles to eventually move the chromosomes, and we're going to see those become evident in the S phase. And then the G phase, or the second gap, here this is when organelles make copies of themselves and the cytoskeleton starts to break down. We're getting ready. We are growing, but we're getting ready to have enough cellular contents in order to take that one cell and turn it into two. We're then going to enter into the mitotic phase. Mitotic phase has two steps. The first is called mitosis. Mitosis is when we're going to divide up the chromosomes. We'll still be in one cell, but we're going to divide the chromosomes into two piles that are eventually going to end up in two cells. Then the second step is called cytokinesis. Cyto meaning cell, kinesis meaning move. We're going to move the cell into becoming two parts or two separate cells. And this is when we're going to divide up the cytoplasm. So in mitosis, we divide up the nucleus with its chromosomes. In cytokinesis, we're going to divide up equally the cytoplasm into its two daughter cells. So let's first take a look at mitosis. So mitosis has as part of it 1, 2, 3, 4, actually 4 steps. We'll see there's a pro-metaphase and a metaphase, but we can really put those two together. So 4 distinct steps. The first being prophase, then metaphase, anaphase, telophase, and then we have cytokinesis, which is a separate process. So here we have pictures of each of those. I'm going to, with descriptions, I am going to go through each of the steps and talk about the various events. You can always refer back to this slide. So mitosis begins with prophase, and here's that picture from prophase. What happens in prophase? Notice we still have one singular cell. The centromeres are formed, so this is going to form the spindle for later on. We still have a nucleus, but it's starting to break down. So the nuclear envelope, which is now dotted in green, is starting to break apart. Why do we say that, or why do we need that? Because these chromosomes are stuck here in the nucleus, but we're going to need to divide them up into 2 different cells. So we're going to have to make the the nuclear envelope go away. The organelles, things like the Golgi and the endoplasmic reticulum, are going to move themselves to the edges of the cell because, again, we're going to have to divide those up. That nucleolus is going to go away temporarily. The centrosomes are going to, here we have our centrosomes with our centrioles. We're going to have to move those to opposite ends of the cell. And microtubules of the spindles are going to start to form. So this is going to be kind of like a clothesline that we're going to hang the chromosomes on. And the sister chromatids, so these are the copies of each chromosome hooked together, are going to coil very tightly so that we start to see individual chromosomes. So instead of seeing like just a mass of chromatin, we're going to actually start to see these X-like structures. This is one chromosome, this is its copy. And that's going to happen for all the chromosomes. So in metaphase, this is the next step. And remember, there was a pro-metaphase and a metaphase. I'm mostly concerned about you just realizing that these are the important steps of metaphase. Those sister chromatids, so these are sister chromatids. This is one chromosome and its copy, are hooked together by a protein called a kinetochore in the centromere region. In this centromere region of the chromosome, and this is what is going to attach to each one of those spindle fibers, the clothesline. So during metaphase, in that centromeric region, that kinetochore is going to attach for each chromosome pair, each sister chromatid, onto a spindle fiber. Oops. Then we go to anaphase, and here's our picture for anaphase. And in anaphase, you'll notice something different here, that the sister chromatids have split apart. They are no longer connected, and they're actually moving towards opposite poles. So those cohesion proteins, which were part of the kinetochore, they degenerate, allowing the 2 sister chromatids to separate. And they each sister chromatid moves in an opposite direction towards its centrosome, and they're pulled by these spindle fibers. The cell gets elongated, forms kind of an oval shape, and we move into our next phase. So by doing this, by separating those in anaphase, we are creating 2 piles of identical populations of a full chromosome complement. So now as we move into telophase, those chromosomes, and if say this organism had only 4 chromosomes, 2 chromosome pairs, we would see, I have the 4 chromosomes that I need for each of the new cells, and I have them in 2 distinct piles. So the chromosomes reach the opposite poles and they begin to decondense. So we're going to unravel them so that you won't see them as a distinct structure anymore. They'll be kind of just a blob. The spindles start to depolymerize or fall apart, so we don't need this blue spindle apparatus anymore. The nuclear envelope starts to reform around the chromosomes to corral them in, and the nucleosomes come back. And we can move on now to cytokinesis. I'll just go back just a moment. You'll notice that the cell is still elongated at this point, but as we enter into cytokinesis, we're going to start to see a little bit of an indent. So in animal cells, we see that an indent starts to form. In cytokinesis, it's called cleavage furrow. And then in here, we form this contractile ring, which is going to eventually break the cell into 2 as a pinching in. We're going to pinch in and divide into 2 cells. Whereas in plant cells, because they have this cell wall, they can't really pinch in. So once we have our chromosomes separated into 2 sides, then as we enter into telophase and reform our nucleus, we're going to start to form a cell plate. And this cell plate is going to grow from the middle to the ends, eventually separating the 2 cells. So animal cells and plant cells have a different cytokinesis. So now I'm going to move on and talk about controlling the cell cycle, because remember, every time we go through the cell cycle completely, we're going to end up with 2 cells. And we don't always need to be dividing our cells. So there are external triggers, there are external forces which can cause a cell to go through the cell cycle. And there are external triggers that can stop that going through. So what are some examples? Well, if we have death of nearby cells, like you get a wound, a cut, like a paper cut, and there's a space there. Now those cells aren't touching one another anymore. They can't communicate, and there will be a signal that, oh, we need to make more skin cells to fill in that paper cut. The release of growth hormones can trigger a cell to go through its cell cycle and make more. And cell crowding can actually inhibit the cell cycle. So some cells, when they touch one another, they send a signal that says, don't go through the cell cycle anymore. We have plenty of cells. There are also internal factors that can regulate the cell cycle. So let's look at some regulation at internal checkpoints. So a new cell must duplicate the original cell. So it must be exactly the same. So that's really important. A mistake, such as a mutated chromosome or the wrong number of chromosomes, we can regulate this at 3 checkpoints in the cell cycle. So we have checkpoint 1, G1, we have checkpoint 2 in G2, and we have a third final checkpoint in the mitotic phase. So the first checkpoint is at the end of G1. The second checkpoint is at the end of G2 when we're ready to go into mitosis. And the third checkpoint is in metaphase of mitosis. So these 3 checkpoints are really important to check and make sure that the cell has everything set up and is ready to successfully go through mitosis and produce 2 new cells. So here we have checkpoint 1 at G1. So the important thing about this checkpoint, we're going to determine whether all the conditions are right for this cell to go through cell division. So external things like growth hormone, adequate cell reserves and nutrition, and the size of the cell, these are really important for making sure that the cell is ready to go through, all the way through mitosis. Also, there's a check to make sure that there's no damage of the DNA as it stands. And a cell that doesn't meet all of these requirements will be halted and not allowed to enter and go through the S phase. So if we don't have everything in line, we're not going to go through the S phase. So you might say, okay, well, what if we don't meet all these conditions? The cycle will get stopped and there'll be an attempt to fix the problem. Like, for example, if the DNA is damaged, can we fix the DNA? There's also an option to enter that G0, that little waiting side step. And for example, if the cell doesn't have sufficient nutrients or the substances it needs to go all the way through, then it will go into that G0 and wait to see if the conditions get better. Okay, the G2 checkpoint. So remember, this is at the end of the G2. So this checkpoint prevents entry into the mitotic phase if certain conditions are not met. So you can't go through G2 unless certain things are ready. So what are those certain things? We again check to make sure the cell size and the protein reserves are good. Do we have everything we need to go through mitosis? But the most important thing of this checkpoint is to make sure that all the chromosomes have been copied. Now, this isn't part of G1 because we haven't copied them yet, but as we progressed from G1 through S, the chromosomes were copied. And this G2 checkpoint wants to make sure that we have copied all the chromosomes properly and that in that copying, none of the DNA has been damaged. If any problems are found here, we're going to halt the cell cycle again and see if we can complete the DNA replication, the copying, or repair any of the damaged DNA. Okay, if everything was good in G2, we progress on into mitosis. And remember, there were those steps in mitosis of prophase, metaphase, anaphase, telophase. We have a checkpoint in metaphase. It occurs near the end of metaphase. And during this, we want to make sure that all the sister chromatids are properly aligned on the spindle fibers. Remember, and this is they attach via this kinetochore in the centromeric range. We don't want them attaching in other places. So are all the sister chromatid pairs attached on the spindle fiber correctly? Because if they're not, then when the kinetochore dissolves and they get pulled to opposite ends, then you will have mistakes. So the cycle will not continue on until all the kinetochores of each pair are firmly anchored to at least 2 spindle fibers. Failure to do this could lead to something that's called non-disjunction, where, say for example, this is not properly attached, and when anaphase happens, then maybe both of these pairs get pulled. Both of these chromosomes get pulled to one side, and the other side gets none, doesn't get a copy of this chromosome. And so the genome in one of those cells will end up with an improper number of chromosomes. So there are molecules inside of cells that help regulate the cell cycle. They are called positive regulators or negative regulators. So positive regulators help you move from one step in the cycle to the next. And some examples of these are called cyclins or cyclin-dependent kinases or CDKs. Negative regulators will stop the cell cycle. And so examples of that, retinoblastoma protein or RB, P53 or P21 are example of molecules that will stop the cell cycle. And it's important to stop when you need to stop. So here are these cyclins or cyclin-dependent kinases. These are positive regulators. These cause the cycle to go forward. And you notice that as you go from G1 to S to G2 to mitosis, the levels of various cyclins are going to change. So the internal and external signals, these will trigger increases in cyclin protein levels. So various messages from inside the cell and outside the cell will cause various of these cyclins to increase, and as say we're going through G1, the cyclin D level goes up, and then cyclin E, and that's going to push us from G1 into S. And then during S, we'll see other cyclins going up, and that will push us from S to G2 and on to mitosis, etc. So how do these positive regulators work? Well, we see that there is a cyclin, and then there's a cyclin-dependent kinase. They're separate. But when we have a, say, phosphate donating protein attached to the kinase, so this is a trigger, we will see that the complex gets phosphorylated, then a target protein can join on, and then this activated protein here will advance the cell cycle. A negative regulatory molecule, so the best ones that we understand right now are the RB, the retinoblastoma protein, P53 and P21. These mostly act at the G1 checkpoint. So we take a unphosphorylated RB, it's going to bind to a transcription factor. So here's the RB. It's going to bind to a transcription factor like E2F, and the E2F cannot then bind to the DNA, and transcription gets blocked. But as the cell grows, we see that as the cell gets bigger, 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 we're going to use ATP to ADP, and we're going to be able to phosphorylate this protein, this retinoblastoma protein. It'll release from the E2F, and then the E2F can bind to the DNA, turns on gene expression, and then we move through the cell cycle. Okay. So cancer and the cell cycle. So when we talk about cancer, we're talking about uncontrolled cell growth. So this was one of those things I said you need to make sure you don't have too many cells growing. So often cancer begins with a gene mutation that results in a faulty protein, a protein that is not working right, and that upsets the cell reproduction, it upsets the cell cycle. Tumors will result when reproduction of mutated cells surpass the growth of normal cells. So a tumor is when these cells just keep growing and growing and growing and piling up. So there's a class of genes which are called proto-oncogenes. These are normal genes. These are normal genes, and they code for positive cell cycle regulators like the cyclins and the CDKs. So what happens is that when these genes get mutated in certain ways, they can become oncogenes. So you can turn a normal proto-oncogene into an oncogene, and an oncogene will result in abnormal cell growth. So for example, if CDK becomes activated without binding to cyclin, normally these two have to bind in order for this process to happen. If it becomes activated without binding to cyclin because of a gene mutation, this will lead to unrestricted division. This binding helps to regulate tumor suppressor genes. Are segments of the DNA, parts of the DNA that code for negative regulator proteins. So remember, DNA codes for proteins. One of those proteins is p53. That's a negative regulator. So when we have a normal p53, if there's DNA damage or cell cycle abnormalities or hypoxia, lack of oxygen, p53 will step in, arrest the cycle, stop the cell cycle, repair the DNA if it can, restart the cycle. If it can't, then that cell will go through apoptosis or programmed cell death, and that damage won't be a problem. But if the p53 gets mutated or broken through a gene mutation, it's going to make an abnormal protein. And when these same DNA damage or cell cycle abnormalities occur, this p53 is no longer able to stop the cell cycle. Cells will continue to go through the cycle and make more and more of themselves. These cells can then become cancerous and form tumors. A mutated p53 doesn't stop the cell cycle. An example of this, it occurs in cervical cancer. Okay, I want to take so far we've been talking just about eukaryotic cells. I want to take just a couple slides and talk about prokaryotic cells. These are bacteria. Prokaryotes, like bacteria, go through a division step called binary fission. So binary fission is much more simple than mitosis and cytokinesis. This is the only way they can produce new individuals. It's very rapid, it's very quick, and it's very simple as compared to mitosis and cytokinesis. So when we look at binary fission for prokaryotes, remember they're single-celled bacteria. They have a DNA molecule which is circular. And what happens is that they essentially just, and it's not in a nucleus, they essentially just make a copy of their DNA. So now they have two copies of their DNA, their circular DNA. They start to pinch in the center. They continue to pinch in the center, and then they form two new bacteria cells. They can accomplish this in about 20 minutes when it takes a full 24 hours for a eukaryotic cell to go through its entire cell cycle. So these bacteria can go through much, much quicker. Okay, that will conclude our Chapter 10 material.